All right, good morning, and thank you for joining us this morning for Winship Grand Rounds. If you're an Emory University or healthcare employee and would like to receive a CME credit for attending today, the login information can be found in the chat feature on the bottom of your screen. If you have any issues with this webinar or with the CME login, please send Julie Hawkins an email or drop a note via the chat feature. So this morning, we are very excited to welcome Dr. David Tuveson. It is truly an honor to host him this morning. In terms of his background, Dr. Tuveson received his bachelor's degree in chemistry from the Massachusetts Institute of Technology in 1987 and his doctoral and medical degrees from the Johns Hopkins School of Medicine in 1994. He completed his residency in internal medicine at the Brigham and Women's Hospital in 1997 and completed a fellowship in hematology and oncology at the Dana-Farber Cancer Institute, Harvard Cancer Center in 2000. He was also a postdoctoral fellow at MIT from 1997 to 2002. So currently, Dr. Tuveson is the director of the Cold Spring Harbor Laboratory Cancer Center in Cold Spring Harbor, New York, where he is also the Roy J. Zuckerberg Professor of Cancer Research. Additionally, he's the chief scientist for the Lustgarten Foundation in Woodbury, New York, the largest pancreatic cancer research philanthropy. Dr. Tuveson is a medical staff affiliate at Memorial Sloan Kettering Cancer Center in New York and works closely with the Cancer Institute at Northwell Health on Long Island. Dr. Tuveson serves on the board of scientific advisors of the National Cancer Institute and was named president-elect of AACR in 2020. He is a world-renowned physician scientist whose basic and translational research focuses on increasing our understanding of the biology of pancreatic cancer. His work has really inspired novel approaches in both preclinical and clinical settings to better diagnose and treat this recalcitrant disease. And in my view, he's one of the true thought leaders related to understanding cancer-associated fibroblasts, especially in the setting of pancreatic cancer. Our own lab is also incredibly grateful for his true collaborative spirit in sharing advice over the years and many critical reagents so that we as a community can better study pancreatic cancer. So welcome, Dr. Tuveson, and we're very much looking forward to your seminar.
Okay, thank you very much, Greg, for that very generous introduction. And good morning to all of you in Atlanta. Thanks again for the invitation. It was some time ago. I don't know if it was during the pandemic or not, but it was a long time ago when we all thought the pandemic would be over by now. But it's not. And I'm hoping that you all are helping to make the pandemic go away by not just talking to your colleagues, but talking to your family and your neighbors and everybody else so that people get the vaccine and keep wearing the masks and behave, you know, so that we can get past this. Okay, so today I'm not going to talk about the pandemic. I'm going to talk about pancreas cancer that has been endemic and relentless, probably since the existence of humans. It's a cancer that we know a lot about now, and I'll tell you a little part that you know, sort of my view of this disease, and encourage all of you to think about how you can work on this disease with Greg and others at Emory if you are still able to change cancer types. And if not, the questions we have in pancreas cancer potentially will apply to your type of cancer. And so again, the title is Overcoming the 7 Deadly Hallmarks of Pancreas Cancer. It's actually the title of a review article that I've promised some editors that I was going to write, and I will still write it. It's not quite done, but you'll see the vague outlines of it. This is Cold Spring Harbor, where I am a faculty member, as Greg mentioned. It's on the North Shore of Long Island. This is autumn when it's quite beautiful. We don't have any leaves on the trees right now, though. Okay, so I'm going to use my laser, and I'm going to move forward. This is my disclosure slide. It's not going to be pertinent for the things I say today, but there is one other disclosure, which is if you hear a small dog barking in the background, that's not my stomach growling. I do live on the water, and so when he sees a bird, he gets a little crazy, but I don't have a backup plan today, so just bear with me. These are the 7 hallmarks of cancer, and it's assembled in this fashion, and I will briefly discuss each of these topics. This is when I took a 2,000-foot view of the whole problem. I think for an AACR meeting I gave lecture at in the fall, I was trying to come up with a simpler way to describe the disease, and this is what came out of that. My view hasn't changed since that time. I think all 7 of these components conspire to generate and support this disease process and make it difficult for patients to get better from it. So I will again touch on all of these things today. The first topic is chronic inflammation. When you think about inflammation, you think about poison ivy, or you think about arthritis, maybe gastritis. Inflammation of the pancreas is called pancreatitis. Many of you know that. And so the pancreas is a non-proliferative tissue that has 2 jobs: digest food, balance blood sugar. Has a few other jobs too, but those are the main 2 jobs of the pancreas. Those functions are made by cells that don't divide. There's also another cell type in the pancreas, the ductal cells, where the digestive enzymes, which are proenzymes, transit through this digestive gland and get into to the duodenum. Also a non-proliferative cell. Proliferation is instigated in the ductal cells and the acinar cells when there's inflammation. Inflammation in this case means the acinar cells have, instead of sending their proenzymes down the ductal tree, have decided to auto-digest. So the cells actually solubilize themselves. And when that happens, you get tissue damage instantly, which attract in immune cells and activate resident fibroblasts in the tissue. And so this issue of chronic inflammation, we all have it. Most of us don't know it. It's subclinical. We have it due to bad luck when gallstones, which are quite small sometimes, go backwards, retrograde up your pancreatic duct instead of from your gallbladder into your intestine. Or you can get it due to exogenous toxins such as alcohol, and then a variety of other reasons, trauma, other medicines, for example, scorpion bites, this and that. Pancreatitis, when it's clinically apparent, it hurts. People hurt. It's actually a more common disease than pancreas cancer. Pancreas cancer is roughly 50 to 55,000 people a year in this country. Pancreatitis is more like 250,000 people a year. So studying pancreatitis has been important because if you have a non-proliferative tissue, you have to become proliferative to become a cancer. And inflammation, in my mind, is how this happens. And so chronic inflammation of the pancreas is caused by many things. First, acute inflammation has to happen, and I describe how that can occur. So 2 recent papers that came out that suggested there were unusual mediators of inflammation in the pancreas are listed here. One is glycosylation. And so we study glycosylation in our laboratory. We have studied it as a way to make better biomarkers of pancreas cancer. And what I mean by that is the 7th deadly hallmark of pancreas cancer. We have no method for that. We have no mammogram, no colonoscopy, no PSA level, no pap smear, nothing. And we do have a blood biomarker though when you have pancreas cancer, which is a carbohydrate antigen called CA19-9. And we sought to use CA19-9 as a way to make better biomarkers in mice. And so we had to put 2 enzymes into mice that are missing fucosyltransferase 3 and beta-3 GALT5. And so we humanized the mouse glycome, as it were. When we did so, we made CA19-9, but we made a whole lot of pancreatitis. And it totally knocked us off our rocker because we hadn't expected any of that. But using mouse and organoid models, we were able to begin to understand this. And it was quite fascinating because aberrant glycosylation, which occurs in every cancer type, in pancreas cancer, the aberrant glycosylation, which is the biomarker of the disease, stimulates proliferation by first causing inflammation. And so that was actually probably one of the most surprising results we've ever had in our laboratory. And Danny Engel, who did all this work, is now leading her lab at the Salk to dig into that. The other observation about inflammation in pancreas cancer is that obesity causes inflammation. And so Mandar Mazumdar had a really nice Cell paper not that long ago, showing that fat mice, genetically fat, they had the leptin axis dysregulated. They actually got pancreas cancer much faster because their islet cell dysfunction occurred, resulting in an increased secretion of cholecystokinin, which some of you will know, cholecystokinin analogs promote inflammation in the pancreas of a mouse. We use it as a drug. We call it cerulean. And so Mandar's paper was, quite amazing because it showed that one of the known risk factors for developing pancreatitis and related to pancreas cancer actually does it through a molecular mechanism that makes sense. And so these were two very recent findings that, have suggested that there are, things we should be thinking about in the prevention space, but also in the intervention space. And so, in Danny's paper, we realized that if we sequester CA19-9, we could actually stop pancreatitis. And therefore the concept is, well, what if you sequester it in pancreas cancer? And so at Cold Spring Harbor, we filed a patent on all this stuff. And a small company in San Diego held the rights to the humanized antibody. It was bought up by this sort of medium to large biotech in Germany called BioNTech. And I met the CEO and all that stuff. And he said, we're going to do this trial in pancreatitis. And the pandemic hit. And BioNTech, as you all know, is the company that made Pfizer's vaccine. They've come back to us recently and said, they're not going to just be a vaccine company. And they still want to do this trial. So I'm going to hold them to their word. I'm going to make my own bloody antibody. But I am pretty hopeful that this is actually going to be a relevant way to intervene in inflammation and maybe in the cancer. So I'm not going to show data for those two things, but these were two provocative things in this space. The next one, though, is desmoplastic stroma, and Greg, in his overly generous introduction, talked a little bit about this. The pancreas cancer is odd in that, look at this little picture over here on the left. The cancer cells are obviously these purple splotches here with the lacunae where the ductal-like structure is. This blue material in this Meson strichrome stain is obviously collagenous material. That's the extracellular matrix. And you look at this, and it's 80–90% blue and 10% purple. And so 8 years ago, when folks were trying to sequence tumors, they gave up on pancreas cancer because they could never get a high enough tumor cellularity. Well, this is true. This is how this cancer lives. It likes to live in a funny way. And so it has a lot of extracellular matrix, and in the matrix live the stromal cells, be they immune cells, be they fibroblasts, be they blood vessels, fat cells, nerves, you name it. And so it's just an odd feature of it. And some years back when we were using the first mouse models of pancreas cancer, we realized that the autochthonous or genetically engineered mouse model didn't respond very well to the medicines we used in the clinic, namely gemcitabine. But if we took the cells out, the cancer cells out, and put them under the skin of a mouse, it responded great. And so we started a process where we realized that the blood vessels, which are here in this blue matrix, were compressed and few in number. And this was part of our early inroads into studying the treatment of this disease, that it was a hypovascular tumor. And that the stroma could modulate responses to just the medicines you try to deliver. There are other aspects to the stroma too, besides drug delivery. There's the aspects that the stroma promotes the survival of cancer cells, for example. And so we study it in mouse models. It's hard to study it in patients. The question is, could we make better models to study it? And we were very fortunate because when we learned how to generate organoids of pancreas cancer, which is a tissue modeling method that you've all heard of by now, where you can take epithelial tissues from rodents and other animals and humans and generate three-dimensional cultures of them, be they from normal tissues or any stage of malignancy. They usually proliferate as proliferative structures. They have a progenitor cell type biology. They're stem-like. You can get them to go to a differentiated state by altering the conditions of the culture. When we made the organoids from pancreas ductal cells, so this is from a mouse, and they basically look like this. If you take the culture, you grow it in a three-dimensional matrix like collagen, or you could use a commercial product like Matrigel, which is extracellular matrix preparation. You could stain it with H&E. But if you include pancreatic stellate cells, which again are a fibroblast population that's resident in the pancreas. It's also resident in the liver. Its job is to store vitamin A until the tissue is antagonized. And antagonized means damaged, inflammation, for example, viral infection. The stellate cells then become activated and they turn into fibroblasts. And when in that process, they get rid of their lipid contents and the vitamin A goes on to become retinoic acid and actually, is very important, obviously, for vision, et cetera, and for all of the retinoic acid receptor biology in your body. This cell population, when we got it out of mice, immortalized them, incubated them with the organoids in particular conditions where we don't have TGF-beta pathway inhibitors, we generated stroma, as you see here, you see in this purple stain, in the H&E. You can also stain it with Mason's trichrome to the right. And so, again, you can almost recapitulate stromal production in a dish in a drop of Matrigel. And by doing so, we stumbled into, again, an area where we could ask, how did this happen? And how it happened was the stellate cells became multiple types of fibroblasts that we could type in different ways by gene and protein biomarkers. And we hypothesized that there were two types of fibroblasts, myofibroblasts, which we called myCAFs, and inflammatory fibroblasts, which we called ICAFs. The myofibroblasts are like the card-carrying cancer-associated fibroblasts. They make collagen, they secrete it, they respond to TGF-beta, they have high levels of smooth muscle actin expression and other TGF-beta target genes. The inflammatory fibroblasts have a different fate. They're the second sister. They are secretory cells. They secrete cytokines, chemokines, growth factors, low-level activation of TGF-beta pathway. And they're distant from the cancer cells. And so the myofibroblasts stick to them. The inflammatory fibroblasts are always distant. And so this was an observational, paper that we had a few years back. And that led to deep mechanistic papers. And one example is shown here, where we actually were able to show that there were ligands coming from the purple cancer cells that could convert stellate cells to either myofibroblasts in blue or inflammatory fibroblasts in yellow. These are two of the ligands, TGF-beta itself, and the various proteases need to activate pro-TGF-beta made by the purple stellate cells. And again, TGF-beta is a totally fascinating cytokine to study. It's actually, again, called transforming growth factor beta, and it does so many things. But in this space, in non-transformed fibroblasts, what it does is it very much polarizes them towards a extremely myofibroblast fate. It's again a fascinating ligand because it doesn't move far. It sticks in the matrix, needs to be processed properly by various proteases, binds like crazy to its receptor with picomolar affinity. There's no, there are gradients of TGF-beta in biology, essentially, almost like it had a lipid tail. So if you do SMAD, phospho-SMAD signaling, you see just like this sort of rim of phospho-SMAD, and you go like 2 cells more, it's dead. IL-1, though, the other ligand made by the purple cancer cells, can just sort of merrily, freely diffuse pretty far away. And the IL-1 is what triggers other fibroblasts to become inflammatory fibroblasts, which is this sort of positive feedback loop, which Julia Biffy was able to identify and interrogate, where the IL-1 receptor activated the expression of various genes which themselves activated JAK-STAT signaling, including LIF. And so this was essentially a mutual antagonism loop because it turns out TGF-beta shuts off the IL-1 receptor. And so it's almost like a developmental biology pathway, which is likely true in developmental biology that, again, cancer has borrowed. And so these were the two types of fibroblasts that we found in this first study. And this became actually quite interesting because our colleague Doug Fearon had actually identified that chemokine CXCL12 is made by fibroblast populations in the pancreas. And in his laboratory work and clinical work, he's shown that this chemokine, although it is a potent molecule in immune regulation, has a effect of making T cells drunk when they see other chemokine gradients. What I mean by that, in some work that Doug has, I think, in press, or maybe he's published it by now, is that when you have a chemokine gradient that T cells normally respond to, if you have CXCL12 present, the T cells are blind and they just are aimless, almost Brownian in how they run around. So CXCL12 does many things. In the bone marrow, does something very special at epithelial cell surfaces. And Doug's done clinical trials to block the receptor for CXCL12, which is CXCR4, and has some really provocative data, about this. So we were interested because CXCL12 was made by ICAPS. And so we thought maybe ICAPS are the cause of immune suppression in pancreas cancer. And so we started to do studies to look at that. And so we used a tool compound, a JAK inhibitor made by AstraZeneca. And you'll see here in these slides that vehicle-treated KPC mice, which is a genetically engineered mouse model for pancreas cancer, they have this much stroma. And if you treat them with a JAK inhibitor, you got a lot more just by Mason strichrome. And you can look here at phosphohistone H3 as a kind of poor man's surrogate for proliferation. Proliferation is shut down in the cancer cells as well as in the stroma. And again, that's because the darn inhibitor, of course, hits every cell type. But the point of this was that we get more stroma when we use the JAK inhibitor. When you look at the myofibroblasts and inflammatory fibroblasts in the setting, the amount of myofibroblasts here goes way up, and the amount of inflammatory fibroblasts goes way down. And the amount of CXCL12 correspondingly goes way down, and they're made by the inflammatory fibroblasts. But look at this stuff on the right. So this has not been cited or published yet. When you do the treatment of vehicle compared to JAK inhibitor treated mice in their pancreas tumor now have a ton of T cells. And it turns out they're both CD4 and CD8. However, if when you use a JAK inhibitor, you prevent a CD8 positive T cell from actually generating an interferon response because you have actually poisoned parts of the T cells that you need for the T cells to do their job. And so this was actually very provocative and suggested if we could hit the inflammatory fibroblasts, maybe we can get real T cells in there. And maybe it was through the CXCL12 axis or other things that inflammatory fibroblasts do. And we're pursuing all of those things quite avidly. Another point though, hearkening back to the drug development vignette that I told you before, the drug development story that we had really relied upon our serendipitous finding that a smoothened inhibitor made the stroma shrink, the blood vessels expand in our mouse model, and we can get more chemotherapy into those tumors and kill more cancer cells. Those mice lived for a couple of weeks longer. They all died in the end. And when they died, their stroma had come back, their blood vessels had gone back down. That went right into a human clinical trial, and the human clinical trial randomized fashion failed. And when I say failed, what I mean by that is that in the trial, the patients who got the smoothen inhibitor with gemcitabine, as compared to gemcitabine alone, they got sicker faster. Cancer didn't get bigger or grow faster. It's just they got sicker. And so when the trial failed, of course, everyone called me and said, what the heck happened? I said, what did the biopsies show? And they said, what biopsies? And this is actually a common problem in pancreas cancer, true in probably almost every solid cancer study, is we don't really know what's happening when we treat our patients. We hope we know what's happening. We think we can figure it out from liquid biomarkers and all of that. And I doubt we can. We get sort of glimpses from that. We need to have much better ways for following what we're doing in the clinic than we do today. So we need innovation in that space. The reason I giving this big vignette though is we had no knowledge, obviously, about fibroblast diversity or subtypes, whatnot, way back then. And so we went back and looked at this problem again. We could not get the same smoothen inhibitor because that company had basically gone under because of that trial failing, and it got sold to another company, turned it into a dermatology drug to treat basal cell carcinoma, where this pathway is mutationally activated. And so we used a different one. We used LDE225, which is an even more potent smoothen inhibitor. And it turns out, in a study that's now been published, that when you compare vehicle-treated to smoothen inhibitor-treated KPC mice, the number of inflammatory fibroblasts goes up very markedly. And this is just sort of one example from one mouse. And the number of myofibroblasts goes down. And so what we've done actually is, and the hedgehog pathway is on in a subset of myofibroblasts. And so the concept now is you might get better drug delivery with a smoothen inhibitor, but you may have just created a whole bunch more of inflammatory fibroblasts, which again, our superficial understanding of that is we may have made cachexia worse, made immune suppression worse, etc. I didn't say this yet, but the IL-6 in a pancreas tumor comes from two cells. It comes from immune cells and inflammatory fibroblasts. It turns out on a cell-based assay, inflammatory fibroblasts make more IL-6 than do immune cells. And IL-6 is certainly a cachectogenic substance in pancreas cancer patients. So this was interesting that these studies led to new insights. The third deadly hallmark is metabolism. Metabolism is the carbon footprint of the cancer cell. And you think about cancer metabolism as sugar, glutamine, mostly, I would say. And every cell needs sugar and glutamine. There's a lot more to it, though, right? So metabolic pathways are engines. That's our economy. How the cells can stay fit. Proteins turn over all the time. Cell division is only one part of the puzzle. Again, a lot of cancer cells can sit there for a long time. It's really metabolism is the thing that's occurring at in microseconds, and that's where all the action is. And for metabolism, you need energy. But again, the way you use your energy is what matters. And pancreas cancer cells obtain and use their energy in ways that other cells don't. And that's very important because anything different about a cancer cell compared to a normal cell becomes automatically something that you should assess as a potential therapeutic vulnerability for that cell. And so Daphne Bar-Sagi's lab had a really terrific observation some time ago that pancreas cancer cells actually eat the protein around them to generate amino acids. And so I talked about sugar and glutamine as being, required by cells. You need to put it in your tissue culture media or else your cells won't grow. It turns out pancreas cancer cells ignore glutamine, that's basically supplied by the bloodstream. They prefer to get their glutamine from the albumin and the collagen that are in that blue stroma that I showed you on the first slide. And so they just bring it in. They macropinocytose protein to get that amino acid and probably many others. So that's a weird behavior, right? So that is totally abnormal behavior. And that's something funny about these cells. They also have dysregulated redox metabolism. And that's something we ended up studying way back when we were trying to understand how RAS oncogenesis occurred. And so these two processes are very important in trying to understand pancreas cancer. And the final pathway, the one that I will talk about though, is cholesterol. Okay, so cholesterol is something you hear about, your family talks about, we worry about. It's basically a good molecule. Without it, we wouldn't be alive. It keeps your membranes fluid. It's part of really life. And it's a complicated molecule shown here on the bottom. You can make it by generating acetyl-CoA from glucose or glutamine and just sort of stapling it together using biochemical reactions. You will make these 5 carbon compounds called isopentyl molecules, moieties that have pyrophosphate on them. That's a double phosphate, obviously. And you will staple together 3 of them to make a farnesyl pyrophosphate, which is a 15 carbon molecule. You can also make geranyl pyrophosphate, which is a 20 carbon molecule. Now, these molecules are very important for getting the RAS family members to the membrane. And the process of again, generating these 15 carbon molecules, like farnesyl pyrophosphate, also makes these ring structures like squalene, which gives rise to cholesterol. So you can make cholesterol, you can obviously eat cholesterol. So people don't usually think of cholesterol as a very exciting way to study metabolism. But again, the pathway of making cholesterol is how you make farnesyl and geranyl pyrophosphate. It's also the pathway of how you make dolichol, which is required for N-glycosylation, and ubiquinone, which is required for coenzyme Q production and mitochondrial function. And if you're a red cell, that's how you generate the chemical structures that you need in heme. So I'm telling you this story because we looked at our organoid series of normal compared to malignant with every step. So pre-malignant, primary tumor, metastasis. And we got a strip of genes at the gene expression level shown here in red, which corresponded to the mevalonate pathway. Again, mevalonate pathway is sort of this pathway, how you make cholesterol. and all these genes were blazing high. And so when you have a pathway that's up, you think, okay, the pathway is turned on. Let's look at the products and, you know, all the mediators. So one of the mediators is SREBP2, a totally fascinating tethered transcription factor, which is in the Golgi and ER. And when it gets activated, the precursor form shown in the upper block goes away, and it ends up going into the nucleus where it can transactivate genes. And so we asked, is there more cholesterol in these cells? And on the left, you can see normal, pannin, tumor, metastasis, cholesterol level all the same in these organoids. So you don't make more cholesterol. Well, you make anything? Well, you make something called cholesterol ester, really high levels in the metastasis compared to the tumor. So I don't show this on this slide, but I'll show you what a cholesterol ester looks like soon. But basically this beta hydroxyl group gets esterified, oftentimes by oleic acid. And this is the sort of the picture of the whole thing. Cholesterol homeostasis and feedback. You can eat cholesterol. It comes in through the LDL receptors, and it becomes free cholesterol once it gets inside the cell, or you can make cholesterol. Cholesterol itself, just as true of most metabolic pathways, feedback inhibits its own pathway. So feedback inhibits the protease that clips this tethered transcription factor, and also feedback inhibits the rate determining step of the mevalonate pathway, which is HMG-CoA reductase. In fact, cholesterol is a better drug for HMG-CoA reductase than our statins. It's just that you can't really give someone more cholesterol when you're trying to lower their cholesterol. So this is, you know, the homeostasis of cholesterol as it was. What other portion of this homeostasis that I haven't mentioned before is that the cholesterol can be removed from this homeostatic mechanism by esterifying it. And the enzymes that esterify it are called SOTs or ACATs. And the one that's relevant in pancreas cancer cells is called SOT1 or ACAT1. And when it esterifies it, it becomes a cholesterol ester. And a cholesterol ester is totally insoluble. So cholesterol is poorly soluble, 75 micromolar in your bloodstream, like 5 micromolar in your tissue culture dish, because that's all the companies want to pay for when they sell you stuff. But when you esterify it, it falls out of solution, does not participate in this homeostatic loop. And it can be, you know, spit out of cells through the ABC transporters, either as the free molecule, and I think also as this ester, although we're still trying to sort that part out. And these transporters obviously are very relevant for those of you who are in the cancer space, because that's how a lot of our drugs get spit out of cells when we're trying to drug them. And so again, as promised, here's the parent molecule. When it's esterified by oleic acid, it takes on this conformation right here. And this is very greasy, as you can imagine. And you get oleic acid mostly from the outside world, from things you should eat and things you shouldn't eat. So lots of nuts have, you know, oleic acid, eggs and beef, you know, et cetera. And so we asked, did it matter? And so we knocked out SOT1 in these organoids, and when we knocked it out, the cholesterol level went down. And we used something we call complete media and cholesterol media. Cholesterol media is essentially physiologic cholesterol concentrations in organoid media. So this is 75 micromolar. And so when you use cholesterol media, you note that the amount of cholesterol ester you start with is much higher than here. But you can really plummet it by knocking out this enzyme, either way. And so what about the cholesterol levels when you knock out this enzyme? Will they go up? So again, it looks like this enzyme is really the regulator of that ratio of cholesterol to cholesterol ester. And of course, the question is, who cares? So when you knock it out though, here's what the cancer looks like before. And this is just like a simple transplant model. You knock it out, these are like kind of cancers, not really. These are little baby things. Some of it are just like the glob of matrigel you had in the pancreas. And you can quantify it any way you want. The interesting thing is really here. In a dish, you don't see this. They proliferate, not as well as you'll see in a second, but they're viable. But when you do the survival study, even though at a certain time point you got basically no cancer, cancer wins. A couple weeks later, the cancer comes back and kills every mouse. So you ask yourself why? And the reviewers, I don't understand. Like sometimes you put a paper up and you know what the reviewers are going to ask, but my ability to predict these days is just like gone way down. And so the reviewers didn't say anything about this question. But the deal is, when this cancer comes back like this, it's not because the gene got re-expressed. It's not because our gene editing didn't work. And it's not because the cousin SOT2 got expressed. It looks because the freaking ABC transporters are now like expressed like they're the most important gene in the cell. And so this is actually totally fascinating, because some of you will know, who've been treating cancer for a long time, that the upregulation of these the efflux transporters is, like that's an old observation, way before, the more sophisticated stuff that we see today has come out. And it's cool because, we actually have ways to drug those transporters. In any case, here we are. This is the pathway, and you can make it from sugar and glutamine, or you can eat it. And I kind of alluded to I gave you some foreshadowing, early on. How might this work? Well, when you knock it out, the cells in complete media, they proliferate a little less, so in red. But you can rescue this proliferation by including farnesyl pyrophosphate in the media. Yeah, somehow it gets across the membrane with its, 2 phosphates and farnesyl. And in the high cholesterol media, where again, the proliferation is much worse than here, you rescue it partially. That's kind of cool. What about the Western blot, the things all of us believe? So when you knock out this gene, SOAT1, and you compare it to the wild type, and just compare lane 1 to lane 3, the amount of membrane-bound RAS, you don't need to, be a sophisticated scientist. It's really low. It's like a third, a quarter, but the total RAS content is the same. You rescue that by putting farnesyl pyrophosphate back in the media. There's an effect in Rho. It's maybe not so impressive on this blot. There's less here than there is here, but you can't rescue it. And that's cool too, because Rho doesn't necessarily only rely on farnesyl, it needs geranyl. So anyway, this is kind of our molecular mechanism, and it's our metabolism story. And so we think that SOAT1 is like a partial synthetic lethal, if that's proper grammar. And it maintains the hyperactivation of RAS through the mevalonate pathway. And so you have a cell that's minding its own business, has homeostasis of cholesterol, and if you knock out SOAT1, the cell could care less. You have a cancer cell, a pancreas cancer cell, and it needs a lot of RAS on the membrane, as you'll see in my next story. It loves RAS, it's addicted to RAS. And so one of the ways it's like kind of used epigenetics to get more RAS is it's turned on this enzyme SOAT1. And you get more RAS and Rho to the membrane by doing so. And you activate that, so you remove the homeostasis. And if you knock it out, it's bad for the cell. And so, the implications is this is a drug target. Turns out you knock it out in mice, the mice make more mice. So the mice can tolerate it. SOAT2 is an enzyme that is required. That's the one that's in the liver. And so that's kind of cool. And it's an enzyme. So, in theory, it's a drug target. And there's some good people working on it. And now we're trying to get into that space. But point number 2 is the one I want you to think about for a second. Is a general property of cancer cell metabolism the loss of negative feedback due to principal product shunting, which therefore would cause an accumulation of parallel branch products? When I asked Sabatini and Vanderheiden this question, they kind of look at me like I got 3 heads. And then I explain it to them, and they don't want to talk to me. So I don't know if that means this is a good idea, but we're looking at this ourselves, and we're not metabolism people, but we have some pretty cool observations, just like by asking this question. And I encourage you to ask it too in your cancer type. All right, so problem number 4, early metastasis. Pancreas cancer does not own this problem. However, pancreas cancer has this problem in a big way. And so when, I'm a medical oncologist, when I hear from a patient, they always have metastatic pancreas cancer. And they say the surgeons won't operate on me because it's spread to my liver. Usually, although it can spread elsewhere too. These are some of the ways that we think about metastasis. We think about the subtypes of pancreas cancer. So pancreas cancer has a RAS mutation 90-95% of the time. The rest of it is wild type for RAS, has other oncogenes. We know most about pancreas cancer by the operations that have happened on the subset of patients who qualify for surgery. And these 3 studies done in 3 totally different places suggest that there's at least one subtype of pancreas cancer based on RNA analysis, so gene expression analysis, that is the outlier of worst outcomes. And so it's blue here, tan here, blue there. And sort of the worst outcome group is quasi-mesenchymal or basal or squamous basal. So you hear these terms, you think EMT. Don't only think EMT. because sometimes you can't really tell based on the shape of the cell. But by gene expression, it has features of mesenchymal cells. Even, still has the RAS mutation and the rest of the tumor suppressor genes that are relevant in pancreas, P53, P16, SMAD4, et cetera. Then there are other colors that don't do as bad, and there's multiple names for them. I'll just simplify this and say there's one other subtype called classical or progenitor, shown blue, and Jen Genier and Rich Moffat's study here. And these other colors in Andrew Bianca's study and Eric Collison's study is red. So the question though, this is the human data. I told you about a bunch of mouse stuff. How are you going to study human pancreas cancer in mice? It's not easy. Koji Miyabayashi was in my lab for 5 years or so. He just went back to Tokyo to start his own lab. And he's here with a black sea bass. I'm not sure that's a keeper, but Koji actually is very careful. He's a gastroenterologist, and he took on a project that others had been unable to move forward with or thought was too insane to try. And the project was to take human organoids and stick them into the duct of the mouse pancreas. So this is the kind of procedure people do in the breast world all the time in mice. It's to put your human breast epithelial cells into a mammary fat pad. Well, no one's really done this in mice before in the pancreas, although the laboratory of Monty Winslow had used this approach to deliver guide RNAs using AAV. And so we talked to Monty and learned his technique for delivering viruses. And Koji was able to learn from Monty's postdoc how to do this properly. And so essentially in non-skid gamma mice, you perforate through the duodenum right here, and then enter into the ampulla, cross-clamp gently, cross-clamp more firmly here in the distal common bile duct. And the idea is to get the organoids up here into the main ductal branches as well as the small ductal tree. And this is called intraductally grafted organoids compared to parenchymally grafted organoids, which is the model that we've generated a long time ago and maybe some of you use. So we compared these two models, and here's kind of the punchline. He can get human pancreatic ductal organoid cells to grow as epithelial cells in the monolayer next to the mouse cells. Here's mouse keratin 19, here's human mitochondria, and this is the basement membrane, here's the lumen. And again, it becomes a, so what? Well, again, the reason we wanted to do this project was to actually study tumor genesis, get a model of early human pancreas cancer. We ended up getting a model though of the subtypes. And so again, in the intraductally grafted organoids, there were two types of models that came out, mice that had a short lifespan and a long lifespan, so sort of a biphasic survival curve, whereas parenchymally, you just got this Gaussian curve. And it turns out basically the mice that didn't live long, the organoids invade rapidly from the duct inside. And if it's slow, they instead do these exophytic lesions that grow into the duct. And this is how they look. Here's a slow intraductal model where basically you see these very pretty frondy structures that grow into the lumen. Here's an eyelid of the NSG mouse. Here's the nice acinar tissue. And here's the meson stain of that. Here's, the cousin, the evil cousin, where it grows fast. And basically you don't see any of this stuff. The acinar cells are gone, the islets are burned out, and the tumor has invaded totally rapidly into the pancreas. These are at, 161 and 102 days respectively. We have all sorts of time points that show this behavior at acute times. And so the question that Koji had initially though was what was wrong, what was different about these two models? So we compared them, and the proliferation rate in the organoids in culture was the same essentially. But you look in vivo and it's not the same. So the fast models had lots of proliferation everywhere. Green again is the human mitochondrial marker. And the slow models only had proliferation where the human pancreas cancer cell met the mouse cell, just right here at the bifurcation. We have no idea of what this means, but it's totally cool. And I'm sure there's some pathway that will be underlying this. And so again, the proliferation pattern is totally different in the slow and the fast models. Well, you had the slow models, and so you could study them, and Koji did. And so you waited for the mice to die, and he could do laser capture on what was in the duct versus what was invaded, and look at the difference in architecture in the duct versus invaded. And invaded, it just looks different, and you could, do your laser capture, do your omics. And he essentially was able to show that the classical subtype of human pancreas cancer was in the duct, and the basal subtype was here in the invaded. And of course I said, well, organoids are polyclonal, so what the heck, you just have like clones that do different things. And Koji says, no, I have made single cell clones and I get the same data. And I said, all right, well, I believe you then. And he did all, clever stuff with, confetti mice and stuff like that, just to show the clonal variety, and that it was an epigenetic process. And so he had this observation, and then we just said, well, could we type the darn samples? And so we had slow on the left and fast on the right. And this is DNA typing, use DNA copy number. And slow basically were diploid for RAS, and fast were, triploid or higher for mutant RAS. And that was, very eye-opening. And so he said, why don't we do cause and effect experiments? That's why we're here. And so he put an ER RAS into these organoids that were slow. And so again, it's estrogen receptor in the N-terminus of RAS. You can do this. It still binds heat shock protein, still gets to the membrane, will do nothing until you add tamoxifen. And when you add tamoxifen, you get these goofy structures. So again, control and then plus tamoxifen. Several cell layers, And most importantly, in vivo, same thing happens. Control, tamoxifen. Well-behaved, metastasis. So it's just crazy. And so, no basal markers, basal markers. Of course, you activate more MAP kinase signaling. And because of this, you can now do, these comparative omic type things. And this is the transcriptomic level of that. And he did it in different models, done in different ways to orthogonally kind of decide upon a set of genes, which were in parallel to this process of progression in pancreas cancer. And when he got to 13 genes, I said, wow, this is crazy. I never would guess it. I was looking for MYC, TP63, the usual suspects of becoming a basal pancreas cancer. I didn't see that stuff. We see genes involved in EMT, extracellular matrix, axon guidance. Genes like kallikrein 6, 7, MMP7, serpin A1, an Adams family member, transglutaminase. And then there are a couple of genes that you say, okay, like PDGF beta, which is going to activate some mitogen. It's going to activate cells nearby in itself. But it turns out that 4 of these 13 genes in sort of single gene analysis of outcomes of patients, which again is, I think speculative way to think about this, all by itself, kallikrein 6, if you had, the 50% of patients that have a lot of it in their tumor, they do terribly compared to the 50% that had less. And the same is true for a couple more genes. And so again, it becomes a question of, well, is one gene relevant? So we knocked out kallikrein 6. And if you knock out kallikrein 6 and do these transplant experiments, here's the tumor before knocking out, here's the tumor after knocking out. You still have a tumor. You still have a cancer cell in a dish. But boy, when you go in vivo, this is like a wimpy cancer. This is mostly like space, fluid. Number of cancer cells is like, 1% on the right compared to the left. And you've knocked out the gene, as shown on the bottom. And so, what does kallikrein 6 do? It's a marinate, right? It just like cuts stuff, cuts up everything. And it digests the stroma and digests the surface proteins and, all that stuff, and releases ligands. I talked about TGF-beta, FGF's classic ligand. It's like sitting there like a, Christmas present waiting to be opened in the matrix. Well, it gets cut like crazy by, enzymes like this. And if you don't release it, your cancer cells are not as happy. And so in any case, there's now tons of like hypothesis directed experiments that can be done because, in our first like N of 1 gene, it looks kind of important. And this is knocking out in the cancer cell and doing these, transplant experiments. So in any case, it looks promising. So we started out by trying to make this model of early cancer invasion. We ended up studying the subtypes of human pancreas cancer. And so, we think RAS activation can, accelerated RAS activation is part of this progression of going from an intraductal neoplasm to a basal-like cancer. And that involves the activation of and the activation by the stroma, involving these processes that I've alluded to. And there's all kinds of hypotheses. Basal pancreas cancer may be the more metastatic one. I have no reason to refute that. And these comparisons may yield new candidates, just like this work started to. And you can ask a lot of questions. Can you prevent the transition from classical to basal? Can the TME by itself, tumor microenvironment, drive this transition? Is pan and classical and pancreas cancer basal? Do they have different cells of origin? I say this as a teaser because there is other groups studying this, not ours so much, and the work is nice. I don't think it's been published yet, but keep your eyes open. But I think it's quite interesting. We now have a model that we can look at therapies because we have these little lesions in the ducts. And we have this model that we can start to do the project Danny was trying to do a long time ago. Look for biomarkers of early human pancreas cancer. And so that's the type of stuff we're doing with that. Okay, almost there. Last science topic, KRAS. You think I've already talked about KRAS, right? I haven't. I talked about ways to get RAS to the membrane. So if I'd been smart 25 years ago, I wouldn't have done any of that stuff with mice and organoids and this and that. I would have just studied KRAS. But I was thick, and so 25 years later, I come back to RAS. KRAS is mutated again in 90 to 95% of pancreas cancer. It's mutated in about a third of lung cancer, about a half of colon cancer, 40 to 50% of colon cancer, and other cancers too. But these are the three main ones that are of most patient volume relevance in our clinics in America. RAS is a fascinating protein. It has, essentially made biology assemble around it so that it can function. There are 3 RAS genes, and the paralogs are expressed throughout the body, although the expression level of each paralog is distinct in different tissues. And then mutations of the RAS paralogs are distinct in different cancers. KRAS itself is the mutated one in pancreas. So it's a G protein, which means it's supposed to cut GTP to GDP and release orthophosphate. When it's bound to GTP is when it binds to other proteins. Those other proteins have something called a RAS binding domain. And the RAF proteins, which again are a series of 3 paralogs, is the classic first described effector of RAS GTP. There are other proteins with RAS binding domains. They are less well understood than the RAF MAP kinase cascade, but that includes the PI3 kinase cascade, for example, includes the RAL GDS pathway, the RAL gaps, and others. RAS, in the normal wild type RAS, it's kind of a bad enzyme. It's very sluggish to cut GTP. It needs extrinsic gap help, GTPase help. And so allosterically, other proteins provide help, either GAP120 or NF1, neurofibromatosis type 1, help it. When you have a mutation in RAS, its intrinsic GTPase function goes down, and its extrinsic GTPase function is lost, because when a glycine is turned to another amino acid, it becomes chiral. That chirality prevents the arginine finger of GAPs from getting in. And so, people think we understand RAS A to Z, but we don't. We understand A and we understand Z, but we don't understand any of those letters in the middle, I don't think. And so I've kind of described all this. This is how you look at the various RAS genes. On the bottom, here's the C terminus, where you have this tail which gets prenylated. And so there's a cysteine followed by 3 more amino acids. The cysteine is obviously where the prenylation occurs. And KRAS4B, which is the splice form of KRAS, which is most pertinent in pancreas cancer, has some differences, has lots of arginines and lysines at the end. So we've thought about RAS a long time. It's just we haven't done anything with RAS other than talk about it. And so we started this experiment a long time ago, before BRA proximity labeling was in vogue. We started with this other enzyme, which the moniker was Apex, where we put a different enzyme on the amino terminus of RAS, and it just caused just chaos inside cells. And so when the biotin ligase proximity labeling method came out, we automatically quickly shifted to that. So the way that works is you put the biotin ligase on your amino or C terminus of your protein, you put it into cells, you add exogenous biotin, and basically the biotin ligase biotinylates what's nearby. And so you can potentially map transient interactions of your client protein. And this is how we created our approach. We took wild type RAS, a mutant of RAS G12D, most relevant one in pancreas cancer. We took the CAX box, which is again the hypervariable region of KRAS4B, and then finally the oncogenic version of RAS G12D, but put in the mutation cysteine to serine at codon 185 so that you couldn't prenylate it. So meaning it would be a cytosolic version of this molecule. We showed in pancreas cancer cells that the cytosolic version was not found at the membrane, but it was found in the cytosol. And we showed that it worked. So when we add a biotin to these cells, we would biotinylate RAF1, CRAF, with the mutant RAS protein and the mutant floating protein, but none of the others. Okay, so then we could do comparative proteomics by IP-mass spec. And this is, again, a Venn diagram of lots of ways of doing that, which, again, the most important, I think, question we asked is, what are the proximal RAS effectors that are at the membrane when you're a mutant for RAS? And this is, again, a way you can plot the data in proteomics, showing enrichment on the left to neutrality over here. And so the enriched proteins that are on the membrane, exclusively on, enriched on the membrane with mutant RAS compared to the cytoplasm are ARAF, RPS 6Ka1, which I didn't know what that was when I saw that, but it's RSK1, SPREAD1, SPREAD2, NF1. I said that as a group because they're known to work as a complex. TORC2, DEPTER, MAPCAP1, Richter. That's all part of TORC2, and Paul Kaveri published that, kind of when we had the first version of our manuscript ready. And so we had to go back and retool the manuscript. So this is what we got, and it was kind of interesting. So ARAF is up high, CRAF is not as prominent, and BRAF is almost neutral. This whole SPREAD2 NF1 is pretty high. RSK is there, which is curious because RSK is a distal effector of the MAP kinase cascade. Gets phosphorylated by ERK, goes in the nucleus, phosphorylates a bunch of stuff, and actually obviously helps run protein translation by being the kinase that activates S6, S6 kinase. And TORC2, again, was just sort of published in the last 2 years while Derek was getting his thesis together. And so RSK became the misfit sock, and so Derek spent really the rest of his PhD trying to figure out RSK. And so this is one of the things he looked at was he confirmed straight away that RSK was biotinylated at the membrane, but not by the floating oncogenic RAS. So it's in this lane and not that. The same is true for ARAF and true for Richter in this experiment. And then he did an interesting experiment. He asked, well, could you pull it down? Could you just IP it? And so again, you can IP RAF bound to RAS GTP, and that's shown here. You can immunoprecipitate using a MYC antibody, RAF1 from the BIR-A G12D RAS because it has a MYC tag on it. You can also biotinylate it equally well. Same is true for ARAF. But these other molecules you can't. You can biotinylate them but not pull them down by IP. That includes RSK1, P110, PI3 kinase, NF1. NF1 has a RBD. And so then it became a question of how the heck does RSK get so near RAS? Because it does not have a RAS binding domain. And so again, this is a dropout screen using CRISPR, and we knocked out all of the molecules which were biotinylated in that top 30 list. And it turns out when you knock out NF1 using CRISPR methods, and it's gone, it's not biotinylated here, and the protein's not there on the other blot, you lose your biotinylation of RSK. And if you go really rapidly to the right, this is done with multiple guides and multiple cell lines. You knock down NF1, NF1 is missing, not biotinylated, RSK is gone at the same time. And this is actually kind of cool because RSK was known to go to the membrane by the really nice paper from John Blennis when you mitogen treat breast cancer cells. He didn't know how it got there, but when it goes to the membrane, it actually inactivates SOS1, a Jeff of RAS, and which suggests it would attenuate mitogenesis. So we've done multiple cause and effect experiments. I will show you the one which I find relevant. When we knock out NF1 in a pancreas cancer cell, you would think depleting a tumor suppressor gene like NF1 would activate RAS more because now the paralogs of RAS, the wild type ones, would have more RAS GTP, and pancreas cancer would be worse, whatever. In addition, you can't tell the difference. You transplant these cells, you knock out NF1, look at these tumors compared to the controls. The tumors are wimps. NF1 is a promoter of pancreas cancer. We think it's through RSK. So the story has gotten far more exciting than the manuscript that is currently under review. And so stay tuned. I think RAS is going to turn out to be, totally fascinating beyond what we learned from flies and, other simple organisms. And, we can't give MEK and AKT inhibitors to patients. Can't do that together. We've done that experiment. We have to think of new ways to drug the system. All right, last two are clinical. I know I'm in the penalty time in the soccer match, and I'll go fast. But some of you are physicians, and this is oncology grand rounds. I apologize. If you have to go see your patients, just go do it. Our patients in pancreas cancer are sick. They are sick. We cannot treat them the way we treat other cancer patients. We cannot say, here are your options. Why don't you go home and think about it, and we'll talk next week. That is all wrong. We have no time to help our patients with pancreas cancer. They have cachexia, they have low physiologic reserve diseases going through their body like wildfire. We got to treat them differently. And so, I don't have a solution for this other than I'm not happy with the way we do it. And I actually am going to try to kick the bucket on this, because I argue with people now in small meetings where I can argue that we're just unethical the way we take care of these patients. So we took our organoid series some years ago, and we asked, could we learn something general? And so Hervé Thiriac, terrific postdoc who now runs the organoid facility at UCSD, took all of the organoids, did the sort of expected omics, but also treated with the medicines in the clinic to ask the IC50 question. Could you profile the organoid and compare it to the gene expression and all of that? And he got so-called exceptional responders. He got this one that responded great to afatinib, a pan-ERB inhibitor, and that has a RAS wild type. It has the ERB2, oncogenic drivers. So we're like other cancers occasionally. But more relevantly, he got these signatures where in blue, you have the signature, in red you don't, and you have mostly blue down, mostly red up. And again, the signature of blue is responding to oxaliplatinum based on the organoids, and the red is lacking that signature. So again, the gene expression of drug response. And this was interesting to us because our collaborators in that study from Toronto, Steve Gallinger, Jen Knox, Grania Kane, and colleagues, had been finding that patients given FOLFIRINOX with pancreas cancer, if they had a basal pancreas cancer, they're in tan, they do terrible compared to those with classical. But if you give them Gen-Abraxane, it's the same. Tan in this case is basal. And so this concept that maybe this was a basal signature, we think it's actually more than that, but it allowed us to actually start an international collaboration. It took all the king's horses to do this, but it's 4 sites in the States and the Canadians, where we have a randomized trial between FOLFIRINOX and Gem-Abraxane, 150 patients with metastatic previously untreated pancreas cancer. They get randomized. We make organoids from all of them. We do the empiric IC50s, but we apply these gene expression signatures to the RNA coming out of either the sample or coming out of the organoid, asking, are the organoid signatures or the organoid predictive of how that patient is actually going to do? Because if they are, that could potentially become a method for how we take care of patients. So we guess up front, right, the chemotherapy. I know chemotherapy is not going to cure our patients, but it will help them. It will buy them time so that as we fiddle around to try to find better targeted and immunologic, et cetera, agents, metabolism drugs, all the things I've been talking about, we give them that time. So that's our approach. Very last topic. I know I'm penalty, commercial time. Diagnostic deficiency. It'll only take 2 minutes. We can't detect it early. I get it. How are we going to get there? So Toby Loba, he's got a terrible fish you don't want to catch. And this was his thesis. It became his passion. He made a lot of antibodies to human pancreas cancer by taking the organoids and injecting various species, alpacas, rats, mice. In the end, we went with the rats. These are a couple of his antibodies. They see pancreas cancer. They don't see the normal human tissues. They don't see other human cancers. So that's kind of cool. And it turns out Toby 85 and 89 see CCAM6, some pancreas cancer version of that. and they don't see pancreatitis on tissue. And this is the spinning mouse experiment. You do the Koji experiment. You can see when you label up these CCAM6 antibodies with zirconium, collaborating with Jason Lewis from Memorial, you can see these little tumors. Jason says you can see them at day 5. Well, he's got better eyes than me. Anyway, this is, these are sub-millimeter tumors at this point. And so, of course, we want to know, do they work in blood? And we're humanizing these antibodies right now. Ralph Ruban's kind of excited about these antibodies. And this is, I think, our first foot in this door. This is a hard door to open. You got to, quote, be better than CA19-9, but I kind of think even just looking for levels of stuff is stupid, but, you got to start there. So these are the 7 deadly hallmarks of pancreas cancer. I went fast. I went over time. I'm sorry about all that stuff. And hopefully I wasn't too casual and you learned something from this. They are filming this so you can play the tape. These are my folks in the pre-pandemic lab. This is Sandra. She now has a fancier job at Brookhaven National Lab. Hans Cleaver's really allowed us to get into this space because he was generous when the lab was in England and decided we would collaborate together, and our first paper was together. We've done more stuff too. He didn't realize he was a Packers fan. Maybe that's not a good thing to say in Atlanta. But this is a fish everyone wants to catch that you all don't have down south. It's called the tautogonitis. A lot of people contributed to this work. They were awesome. Julia, Ella and Jen are the fibroblast crew. Hervé and Dennis are the organoid crew. Derek needs an arrow. He's the guy who discovered RSK. Toby did the biomarkers and SOAT1 with Julia. And the lab is just a terrific place. And many of them are listed here. And the funding, again, comes from the NCI, from the Lustgarten Foundation, as well as the Thompson Family Foundation. And many, many terrific collaborators. Some of them I've mentioned throughout the talk. I think I've mentioned almost all of them. So again, I thank you for your attention. Apologies again for running over. I don't know how they're going to do the question and answer bit because I've sort of gobbled up all that extra time, but I'm here for that. Thanks.
Thank you so much. Julie, do we have a few minutes for questions?
Yeah, go ahead.
Okay, so thank you again. We had one in the chat from Dr. Lawson asking what happens to the basal-like cells when they get to the liver. Do they generate the same sorts of response they generate in the pancreas?
Yeah, so in our transplant models, I don't know that we've looked at that. Well, we do get activation of the stroma for sure. I think that was part of your question. We haven't done a detailed omic analysis though of the metastatic cells in the liver from these transplant models. But we do get stromal activation. So again, grossly at the level of histology, there is an activation, yeah.
Great, and then maybe the last question from an anonymous attendee. Does de novo lipid biosynthesis pathway through ACLY get upregulated in metastatic PDAC to increase free fatty acid for providing substrates for the esterification of cholesterol esters?
Yeah, so that's actually, that's a good question. Yeah, I'll actually, so you can stay anonymous if you want, but if you send me your email address, I'll actually give you a real answer to that. I don't think that we have seen that, but we do see other things in the lipid biogenesis pathways. Yep, we do see other things there. So if you want to talk about it, just send a real email.
All right, I think we need to move on because Dr.
Let me tell you how Greg, how I've done this in the past those places. Yeah. So honestly, if you all have real questions, send your questions to Greg with your email. And then, I don't know tonight, but certainly by like Sunday, I will have answered it. And I'll copy Greg on each thing so he knows that I've actually done it.
I am happy to interface. So,
and anything I've talked about, you can have, like Greg said, any cell, any mouse, any whatever. So don't ask stupid questions, ask good questions.
All right, thank you again so much. We'll give you a little bit of a break here before you have a meeting at 8:45. All right. And thanks for your time. Maybe you can stay on and Julie can end the feed.
Okay, thank you everybody. Have a great day.
And thank you. The meetings are on a separate link. So within your itinerary that we emailed over.
Perfect.
So we'll join you there at 8:45.
Do you have all that, Dave? You know where you're going?
Yeah, I just have to find the email. I'll get it.
Sounds good. I think you got a few minutes to get organized, and we'll see you about 9:45, myself and Dr. El Reyes. So thanks again so much. Great talk.
My pleasure.